Okay, so now after we've finished, talking about all of our vocabulary, let's talk about Gregor Mendel. Gregor Mendel was actually an Austrian monk who's called the father of genetics. So he is considered to be the father of genetics because he's the one that did um, all the pioneering in genetics. He studied pea plants and specifically studied 7 traits in pea plants. And it was really easy for him to um, trace these different traits that he was looking at through several generations because these pea plants were actually had 2 distinct forms. There was a wrinkled form or a non-wrinkled form of a pea pod, or there was a tall pea plant or a short pea plant. He actually didn't get a lot of credit for his work back in his day. It wasn't until after he passed away that people were like, oh, this genetic stuff, there's actually some truth to it. Moving on, Gregor Mendel. So the traits that Gregor Mendel chose to study were easily observable in 2 distinct forms. So there was either, a seed shape that was spherical or dented, or what I like to call wrinkled. A seed color that was either yellow or green, or a flower color that was either purple or white, or a stem height that was either tall or short. The significance of Mendel's work was not recognized until the turn of the 20th century. Its rediscovery prompted the foundation of genetics. So, this is just review from before. We have a big T. That means we have a homozygous dominant or purebred tall. That means that the big T is dominant because we use capital letters to represent dominant, and it's tall. Heterozygous or hybrid, which means that we have 2 different alleles here. One of the alleles is for tall and the other allele is for short. Since the tall allele is dominant to the short allele, this big T dominates over the smaller t, and we have a tall pea plant. Little t, this is actually recessive. Little t, since we don't have any dominant traits dominating over these recessive genes, they're able to be expressed as short. Geneticists apply mathematical principles of probabilities to Mendel's laws of heredity in order to predict the results of simple genetic crosses. And Mendel's laws of heredity are based on his mathematical analysis of observations of patterns of the inheritance of traits. What that means is he was observing specific probabilities. If he crossed a homozygous tall with a homozygous short, he would have 100% tall plants. And we'll talk about these probabilities and why he was seeing that. Now, if he had taken a heterozygous, 2 heterozygous pea plants, he was seeing a 75% chance of tall and 25% chance of short. So the laws of probability govern simple genetic recombinations. To use this, we use Punnett squares. To complete a Punnett square, we use a letter to represent each allele, and we represent the dominant allele with the capital letter, and the recessive allele is given the same letter but in lowercase. For the pea plant flowers, dominant purple flower, big P, recessive white color, little p. If both parents are purebred, then the purple color parent must be capital P, and the white color parent must be little p. So how can we predict these results? We can complete the possible combinations. So we're going to take a homozygous dominant purple flower, and we're going to take a homozygous recessive. Now when we're doing a Punnett square, we put the parents on the side and we put each allele over one of the blocks. Okay? We're going to draw 4 blocks. And here we go. We have big P, little p, big P, little p, big P, little p, big P, little p. And what we're seeing here is we should see 100% purple offspring here from that. And we call this the F1 generation. And because it's the first generation for the cross of the parents. And the offspring are all purple colored hybrids. So they're 100% purple. And what we did here is we just said we're going to take one from the top, one from the side. Okay? So we can use another Punnett square to predict the F2 generation of offspring. And the F2 generation is simply just taking the offspring from the F1 generation and breeding them again. So here we have the F2 generation. Big P, little p, and big P, little p. So there are 2 hybrids, and we're going to cross them. And what we're going to see is we get one big P, little p, one big P, little p, one capital P, one capital P, lowercase p, and one lowercase p, which means that we're going to have this guy is going to be purple, this guy is going to be purple because this big P is going to be dominating over this little p. This guy right here is going to be purple because he's, again, the big P is going to be dominating over the little p. And here we don't have any dominant traits, so our recessive traits are able to show. So, what this tells us is the results are always mathematically the same. A 3-to-1 ratio with 75% purple and 25% white offspring. Now, if you're not a math person, there's a simple way for you to do this in your head. I remember I wasn't too big of a math person, so I just thought of these 4 squares as 4 quarters. And if I have 1, 2, 3 squares, then that's like having 3 quarters, like having 75 cents, or in this case 75%, and 1 quarter would be 25%. Now if we're doing a ratio, we just look and we see 1, 2, 3 purple flowers to 1 white flower, okay? And that's our phenotypic ratio. And remember, our phenotypic, or our phenotype, is the physical appearance of something. So what we can see, what it looks like. And these are purple flowers and these are white flowers, so that gives us a 3-to-1 ratio. While genotypic ratio is a little different, because these are the genes actually present in the organism. Well, this guy is different from these two. He's got 2 uppercase P's. He is homozygous. So we actually have a 1 to 2 to 1 ratio because we have 1 homozygous dominant uppercase P, 2 heterozygous purples, and 1 homozygous recessive. Okay. I'm going to let you guys, I'm going to stop this now.